pollination. Although in the last video we have watched a little about the pollination, but now again pollination, we know that transfer of pollen grain from anther to stigma, which I have already discussed in the last video. And we know that the pollination is divided into three types. One is autogamy, next one is gaetogamy, and third one is genogamy. But auto means self, so conventionally we divide the pollination into two. One is self-pollination and second one is cross-pollination. One is self-pollination and next one is cross-pollination. Self-autogamy is the example of self-pollinations where the pollination takes place within a flower, and if of the same plant, flower of the same plants or flower of the different plants, that is comes under the cross-pollinations, that is gaetogamy and genogamy. Now we know that cross self-pollination is independent, but cross-pollination is dependent. Means cross-pollination it depends on the several agents. So the agents for cross-pollination: these are, one is insect, second wind, third water, fourth animal, and fifth birds. The pollination which take place by means of stomach, by means of insects, they comes under entomophily. Here the main in those flowers which are pollinated by insect, they have certain characters, that is colors, odors, and nectars. These are found in these flowers, and due to that the flower attacks and help in the pollinations. Wind insects, that is anemophily. They are pollinated by the wind in those flowers which these things are absent. So no insect comes for pollination, so that pollination takes place by means of wind, and the winds, the pollens are light in weight, many in number, and also wing-shaped, by which it can move in the winds. So examples: maize, water, actually the aquatic plants, the water help in the pollution in the aquatic plant like Rallisnaria, that is by means of animals and by means and the water is known as hydrophily. Animal that called zoophily, and birds by means of small birds which enter their beaks in to suck the nectar, that's help in the pollination. Ornithophilia. So these are the agents which help in the pollination, cross pollination. And we know that due to cross pollination, the good, viable and strong seeds are produced. Now the consequences of cross pollinations. We know that due to cross pollinations, more strong, viable seeds are produced. And next, the continuation of self-pollination means if the self-pollination is continued, continuation of self-pollination results the inbreeding results in inbreeding depressions. Means continuation of self-pollination is not good for the plants. So flowering plants have many devices to avoid self-pollination and to encourage cross pollination. Means in the flowers there are so many adaptations which avoids the self pollinations and to encourage the cross pollinations. They are also called as outbreeding devices. That is outbreeding devices. One is unisexuality. We know that anisexual flowers are those flowers in which male flowers and female flowers are separately present, and means in male and female flowers separate, so self-pollination is impossible. Self-pollination is not possible. Cross-pollination is possible. Next one, self sterility. Although the flower is bisexual, if the self-pollination takes place and inhibitory substance secretes from the stigma which inhibits the fertilization. In such position, the self-pollination is not successful, only the cross-pollination is successful. So, the cross-pollination is good for the such type of plants. Heterostyle, next one is, in which the flower is bisexual, but there is great difference in the level of anther and stigma. So the cross self-pollination is not possible like that. But this is the flower. This is the female reproductive parts, and this is the stamens. So that's why a great difference between that. So the pollen is unable to reach the stigma. So the self-pollination is not possible. Cross-pollination takes place. Next one, that found in the family Euphorbia. Orchogamy. Flower is bisexual, but there is a partition or barrier between stamen and pistil. So the self-pollination does not take place, and the self-pollination cannot take place. Cross-pollination is possible. Example, calotropis. So they are the devices which are used, which are adapted in the flowers to avoid self-pollination and to encourage cross-pollination. Now after the pollination, next one, pollen, pistil, interaction. Pollen pistil interaction. We know that in the pollinations, the pollen grains transfer from anther to stigma, but it is not essential. It is not essential that by pollination the stigma will get the pollen grain of same species. I mean, it is not essential that all the time right pollen will transfer to the right stigma. If the stigma accepts right, if the stigma accepts right pollen grain, it will promote the post pollination events. Means if the stigma will get the right pollen grains, means of its same species, then it will allow for the post pollination events, means for the post pollination process. And if stigma gets the wrong pollens, then it will be rejected. So a stigma has capacity to identify whether the pollens is of same species or of different species. Means first a stigma accepts the pollens and then recognize or identify it, and it is called pollen pistil interaction. So the first stigma accepts the pollens by means of pollinations and then recognize or identify it is whether it is of same species or not, and so it is called pollen pistil interaction. And in this the chemicals which is secreted by the stigma help to identify. Now the next point is artificial hybridizations. That is hybridization artificially. It is one of the main method of crop improvement planning. It is one of the main methods for crop improvement plannings. In this, only the deserts pollens are used for the pollinations. Only some desired pollens are used for the pollination. In a bisexual flower, the anther is removed. The anther is removed before dehiscent. That is in immature state, and that is called emasculation. That is known as emasculation. So emasculation is a process in which the anther is removed before the dehiscence, means in the immature state it is removed away. In the artificial hybridization, how the emasculation is done. Suppose this is the bisexual flower, this is the stamen, and this is the pistil. Now, by the help of a forceps, we remove these stamens, means remove a stamen by forceps. Now then it is now the after this is a pistil, and it is bagged by butter paper. It is bagging. Now after that, when it got matured, then by open the bag, we powdered the desired pollen grain on the stigma. We open it and powdered the desired pollen grains on the stigma. It is a stigma, and again covered it for the maturations and pollination, for the maturations and fertilization. Means means it is covered again for the fertilization, and post fertilization process, means that is for the formation of seed. Means after removing the this is the flower, after removing the stamen, we bind it by a butter paper, that is called bagging. When it got matured, we opened the bag and powdered the desired pollen grains on the stigma, and again covered it for the fertilization and post-fertilization process, that is for the formation of seed. Such type of seed is the hybrid seed, and which is more beneficial nowadays. Now the fertilization, we know that fertilization is the fusion of male gamete with female gamete. By pollinations, the By pollination, the spore transfers on the by the pollination, the pollen grain come on the stigma. We know that is it is two words. One is exine and next one is in time, and there is a pole, one or two, that is and in this a big nucleus and a spindle shaped nucleus. This is vegetative and this is generative. Now the pollen grains come on the stigma. The stigma secretes a sucrose solution, that is sugar plus water solution, which is absorbed by the pollen, which is absorbed and becomes active. Means the stigma secretes the sucrose solution which is absorbed by the pollens and becomes active and becomes active. Now the vegetative cells or tube cells develops and emerges out through germ pore. This is the germ pore from which this is the pollen tube. This is comes and that. This is the pollen grain which comes out from the pollens, moves towards the ovules or imperial sac. This is the chalazal part. This is nucellus and this is embryo sac, and the genetic nucleus divides into two male gametes. Genetic nucleus divides mitotically into male gametes. Now these male gametes will also come through that. This is the male gamete. Now the pollen tube may enter towards the chalaza side, towards the integument side, and towards the micropyle side. Understand? By any way, but generally it goes towards the micropyle side. But the three chances, one is through the chalaza, second one through the integuments, and third one through the micropyle. So that is and this is the egg cells, this is synergids, this is polar nuclei, and these are antipodals. That's it. Now this is the structure of embryo sacs. So the male gamete will move slowly toward through the pollen tube and comes in contact of the embryo sac. Now we shall see here. This is the egg and these are synergids. One thing you know, this is the egg and this is the synergid. Here is a small apparatus is found, that is known as filiform apparatus. Now when the pollen tubes come in contact of the embryo sacs, the first one is the synergids, degenerates, and the filiform makes passage for pollen tube. Now this is the pollen tube, it comes, it dissolves and provide a passage for entrance. When it enters, the pollen tube dissolves and the first male gamete fuses with egg called fertilization, and that produces zygote. So due to fertilization by male gamete with egg forms zygote, and second male gamete fuses with polar nuclei, and that it forms primary endospermic nucleus, and this is called triple fusion. Means N polar nuclear 2N, and that is triploid. Zygote is diploid, means N plus N, 2N, N plus, here is 2, N plus N, means this is N and this is N plus N. So in this way triploid is formed, that is known as triple fusion, and the combinedly is called zygote plus triple fusion, that is fertilization plus triple fusion, combinedly called double fertilization. And from the fertilization zygote is developed, and from the triple fusion primary endospermic nucleus is formed, which later developed into endosperm. So and it is a nutritive layer. So the zygote and endosperm is formed by double fertilization. Means zygote is formed by the fertilization, and this primary endospermic nucleus is formed by the triple fusions, and which develop into endosperms. And that is the process where the fertilization and double fertilization takes place. And finally, next one, post fertilization changes, which we will discuss in the next video.